All right, so let's see. We've worked through monohybrid crosses. You've worked through some dihybrid crosses, and now you get the chance to look at a couple of non-Mendelian or non-Mendelian traits. We call two of them incomplete and codominance, and we'll look at how both of these types of traits work. They're very easy, so I don't think you're going to have any difficulty with this. I'll start first, though, with incomplete dominance. Or actually, let me take that back. Let's try this first. Let me give you an overview of each of them. Um, when you look at incomplete and codominance, when we look at the traits, there's actually no dominant or recessive traits. So for incomplete dominance, both of them blend and you end up forming a whole new trait. And that's what you're going to see with the heterozygous genotype. So remember, your heterozygous genotype had the capital letter and the lowercase letter. For codominance, same is true. You don't have a dominant or a recessive trait, but both traits actually appear together, sort of with each other. So sometimes, or in most cases for codominance, we'll see the traits as two completely different letters because the two traits will work together and be shown at the same time. So I'll start first with incomplete dominance, and we're going to use flowers to start. The flowers that you see here in this picture, we call these impatiens, but you can see this happen with a number of different flowers where you can have one flower color like red and another color like white blend together and actually form pink flowers. And so the ways that we could write this is we could use a capital F to represent the red flowers. And so if you had red flowers, you would have two capital Fs. White flowers, we could use two capital W's to represent the white. And so for pink flowers, that would be the heterozygous genotype, which means you'd have one of each allele, and that would produce pink. The white and the red together form a whole different color, and that's what incomplete dominance shows. If we were to write this using the same letter, you would write it just like you were writing it before. You could pick one of the traits to be the capital, one of the traits to be the lowercase, and then the heterozygous genotype would represent that new color. The only reason this may not always be the best way to show it is just because we're accustomed to the capitals meaning dominant and the lowercase meaning recessive. And so I don't want you to get confused because there is no dominant or recessive here when you're talking about incomplete dominance. So let's say we were going to cross a red flower with a white flower. Okay, and so we have F's to represent the red flowers, and we have W's to represent the white. Notice when we cross these, you get an F and a W in every one of your Punnett squares. And so what this tells us is we have a 100% chance that all of the flowers will end up being pink because you get one allele from each of the parent flowers. So you get one red allele, one white allele, and that means all of your offspring flowers would end up being pink. If we look at incomplete dominance again, and let's say this time we took all the pink flowers that we had crossed the first time around, and we decide to cross two of those pink flowers. So we have our pink flowers, which is represented by an F and a W, the heterozygous genotype, and we put the other pink flower here. When we cross these pink flowers, you'll notice that what happens is you get this box with two Fs, which is red, this box, an F and a W, which represents pink, this one, an F and a W that represents pink, and then this one, which represents white. And so this time around, with two pink flowers, you actually have a 25% chance of having red flowers, a 25% chance of them being white, but a 50% chance that the flowers could be pink, just like the parents. So just keep in mind with this incomplete dominance, there is no dominant or recessive. You actually end up with a whole new trait that's blended together. This is another really good example. Straight hair versus curly hair. And then you got that in between, which is wavy. It's 3 different phenotypes. So straight hair is one phenotype, curly hair is one phenotype, and then wavy hair is another. So wavy hair is actually a combination of one curly haired allele and one straight hair allele. So if you have wavy hair, it's very possible, or it is very probable, that one parent has curly hair and one parent has straight hair. So let's look at that in terms of a real example. Let's say we cross a guy with wavy hair and a girl that has curly hair. So wavy hair, for this case, because it is the combination of the two, we would use the C and the S, the C representing curly and the S representing straight. And then our curly haired person would have two C's because that's the trait, or the genotype, for curly hair. So here's our wavy haired guy. Here's our curly haired girl. If we cross these two, you'll notice in our Punnett square, we get a 50% chance that our offspring or our children will have curly hair, and a 50% chance that the children would have straight hair. Not straight hair, wavy hair, So let me say that all over again. A 50% chance of curly hair and a 50% chance of wavy hair, just to make sure there's no confusion. Now to jump into codominance, remember with incomplete dominance, like I just showed you, it was all about a blended trait. Codominance is a little bit different. If you look at the chickens here, hopefully you can see right away how codominance is different from incomplete dominance. Incomplete dominance, remember, we got that whole new color. We had red flowers, we had white flowers, we had pink flowers. Now we have two traits that both act as if they're dominant, and so they both appear in the phenotype. So if we have a black feathered chicken, a white feathered chicken, and those two combinations are blended, we can actually get a black and white feathered chicken. So if black feathers is the trait, we could use two capital B's to represent that. White feathers is the trait, we can use two capital W's. And so black and white feathers in codominance would be represented with a capital B and a capital W. So just like before, if we cross a black feathered chicken and a white feathered chicken, you know that you have a 100% chance of producing that heterozygous genotype. But in this case, we don't get gray, we get a black and white feathered chicken, where some of the feathers are black, some of the feathers are white. There's no blending of the two colors like there was in incomplete dominance. The roan cow is a perfect example of how codominance works. We also have roan horses. Roan is the coloring that we call a cow when it has a combination of that reddish coat and the white coat. I want to show you this time, though, a different way of writing some of our genetic traits. You'll notice this time there's R's everywhere, and those R's sort of hold down a little superscript or a tiny little letter that's up in the top corner. So you don't really mess with those big capital letters a whole lot. They're just placeholders for whatever trait it is or whatever organism we're looking at. And the small letters up in the top corner, they are what are going to actually represent the genotypes that are going to give us the phenotype. And so in this case, a red cow would be represented by looking at it this way: a capital R with a little r, capital with a little r. A white cow, we'd still have our capital R's, but notice our little superscripts are both W's. Then your roan cow would be represented by a little r and a little w. If you pulled these big R's away, it would be just like what we had up here before on this slide. Imagine we had a C here to represent that we're talking about chickens. And we have a capital C and a B up at the top, a capital C and a W at the top. So it's basically the same thing, The larger letters here are just holding the place for those traits. So if we wanted to cross a white cow, which has the two little W's, with a roan cow, which has an R and a W, this is what our Punnett square would look like. So when we did this cross, you kind of carry over all those capital R's. For this one, you'd get a little R and a little W, a little R and a little W. And for these, you would get two little W's, which means if you crossed a white cow and a roan cow, you'd have a 50% chance of having roan cows and a 50% chance of having white cows. That pretty much sums it up for incomplete and codominance. Once you've learned how to do Punnett squares, these two types of dominances are very, very simple to understand. Just keep in mind that incomplete dominance is a blending of a trait. So you do get a whole new trait out of the heterozygous genotype, whereas codominance is both traits showing. So you don't get a blend, you actually see both traits. So thinking of things like your spotted cows, those are really good examples of codominance compared to incomplete dominance, where you get a whole new trait in the process.